Assalamu alaikum. Today we will talk about the current issue, that is Omicron. What do we know about the new variant, coronavirus variant, and should we be worried about it or not? The variant was first detected in Botswana and contains a high number of mutations associated with increased immune evasion. So let's have a look on it. COVID-19 variants bring new difficulties, uncertainties in fight against pandemic. Variants of concerns like alpha variant, delta variant, gamma variant, and beta variants. Alpha variant, as we know, was first detected in the UK in 2020 September. There was 43 to 90% more transmissible, and its various functions suggest it leads to higher fatality rate. Because the dominant strain is in the UK, it's played a leading role in serving cases to the winter months. The Delta variants first detected in India in October 2020. It thought to be responding for the second outbreak wave in India as well interrupted in March and raised deadly cases number up to 4 lakhs. Is estimated to be 51 to 67% more transmissible than the alpha variant. There are findings that it reduces the efficiency of vaccines. The high contaminant variants is appearance to soon become the dominant strain worldwide. The Delta Plus mutations that have been in April 2021 has been a cause of concern. The next ranges of the Gamma variant, first detected in arrivals from Brazil in Japan in January 2020. It has been caused 17 amino acid changes in the viruses. The emergence of a new outbreak wave on Manaus, the capital of Brazil state of Amazonas, in January 2020, while the Beta variant was first detected in South Africa in 2020. Observed more commonly among young individuals with no previous history of acute disease and poses disastrous stress. The variant may reduce protection of COVID-19 vaccine and develops resistance to antibodies. A coronavirus variant that has been detected and contains high number of mutations within spike protein, some of which may help the virus to evade a vaccine-induced immune protection. B.1.1.529 is known to contain 32 mutations and it's commonly known as Omicron in the gene for the coronavirus spike protein compared to the original strain of SARS-CoV-2 first identified in Wuhan, China. Many of these mutations are also found in other variants of cassette, including Delta variant, but many others are not. Some of the mutations found in other variants of concern include N501Y mutation, which improves spike protein binding to cellular receptors. It basically improves the binding of the spike protein to the cellular receptors, while the D614G mutation thought to enhance viral replication, both of which could make the virus more contagious. Other includes K4179 and T478K mutations will help the virus to avoid neutralizing antibodies generated to vaccine or previous infection. Balak said, I would definitely accept it to be poorly recognized by neutralizing antibodies related to alpha or delta. It is difficult to predict how transmittable it may be at this stage. But the good news is that diagnostic and disease surveillance labs should be able to detect this variant ahead of whole genome sequencing using a particular type of PCR test, which should make it easier to track and understand its spread. It would also mean that they have a clear idea of how much of a threat it poses sooner, which would enable the governments to take a step up to containment measure if necessary. So let's have a look about the potential impact of mutations. Multiple RBD and NTD mutations associated with resistance to neutralizing antibodies and therapeutic monoclonal antibodies. Cluster of mutations like H655Y plus N679K and P981H adjacent to S1 and S2 furin cleavage site associated with more efficiency cell entry, enhanced transmittability. Basically, it allows the more efficient cell entry by attacking the cleavage sites. NSP6 deletion, similar to deletion to alpha, beta, and gamma lambda, may be associated with evasion of innate immunity. Interferon antagonism, which we called it in the scientific term, could also enhance transmissibility. While the R203K plus G204R mutation in nucleocapsids, which are also seen in alpha, gamma, and lambda, associated with increased infectivity, that is more effectable, infect the more people. So let's have the list of mutations here, and you can have a clear side of this. In the Omicron, we see all of this as we had discussed. Let's have a view of mutations of Omicron 50 with 30 in the spike protein and 15 in its receptor binding domain. As you can see, number 1, which is shown here in the blue, the combination of mutations at K417N to S477N and Q498R and N501Y is thought to be an antibody evasion strategy. While the number 2, which is in circle red color, they show the deletions at position of 6, 9 and 70, mean the variant can be detected using some PCR tests without the full need of whole genome sequencing. The red color shows insertion and deletions, while the blue color show mutations. This is the top-down view. The spike is built of 3 protein chains, and here's the side-on view. The third, which we can see the 4 new mutations at Q339D and S371L and S373P and S375F may create additional obstacle for certain antibodies. The 1 and 2 points are also in the delta and alpha variants, but the Omicron has these 3 new mutations. 3 mutations near the furin cleavage site at H655Y and N679K and P681H may be associated with increased transmissibility. This allows the increased transmissibility of this variant. While the 5 show 3 additional deletions at position L105, S106 and G107 in NSP6, a protein that is not part of the spike, may aid in immune evasions. In the 6th point, we see in total there are 15 mutations in the receptor binding domain, the part of the spike that mediates how easily the virus attaches itself to the cell. So this is the overview of the mutation of Omicron. So if we compare it with the delta, the mutation residue in spike proteins is greater than 60% available sequence. The red color shows 70% the structure of SARS spike protein to active sites is in orange color, while the residue color against different mutation rates. There is 43rd amino acid mutation residue in spike protein in Omicron and 34 residues in greater than 60% of available sequence in Omicron in compare with the delta variant. So next, if we compare it with the delta variants, you can see the point mutations in blue color and insertion deletions in red colors. The comparison to delta makes it more obvious that we are dealing with a new spike protein in Omicron. Basically, there are more spike protein mutations. From faster spread to asymptomatic disease to more severe outcome, everything seemed possible at this point. You can have a better look at this picture. Now we will look at the table, the mutation map of the 5 variants of concern, that is alpha, beta, gamma, delta and Omicron and lambda. We can see the mutation map. This table displays the amino acid mutations shared by the variants below and the other defining mutations of the variants below. You can have a look at this. You can see that the Omicron has all the amino acid variations of alpha, beta, gamma and delta. In addition to this, it has other mutations which are not present in alpha, beta, gamma and delta. It has its own mutations that are in the spike protein mostly. So this is all from now. Thank you. Subscribe for more videos if you like it. Thank you.